Hello everyone, in this video I am going to tell you something about dental plaque. So first of all we need to define what is plaque. Okay, we need to define what is dental plaque. So dental plaque is defined clinically as a structured, resilient, yellowish, grayish substance that adheres tenaciously to the intraoral hard surfaces. Intraoral hard surfaces including removable and fixed restorations. So what is this? This is a definition of dental plaque. So dental plaque is defined clinically as a structured, resilient, yellowish grayish. The color of dental plaque is somewhat yellowish grayish substance that adheres tenaciously, like it adheres to the tooth surface tightly, tenaciously to the intraoral hard structures. Any intraoral hard structure like a tooth, root, if it is exposed, okay, tenaciously to the intraoral hard surfaces, including if the patient is wearing removable or fixed restorations, then dental plaque can also adheres to the removable and the fixed restorations. So this is the definition of dental plaque. Now we need to know in this is formation of dental plaque. Formation of dental plaque, biofilm. So the formation of dental plaque includes three phases. Phase 1 is formation of plaque on tooth surface. Phase 2, initial adhesion or attachment of bacteria, okay, and the phase 3 is secondary colonization or plaque maturation. Now we discuss each and every phase one by one in detail. Okay, first of all, phase 1, which is this. So all surfaces of the oral cavity, all surfaces in the oral cavity, both hard and soft tissues, okay. Oral cavity includes some hard tissues and some soft tissues, both are included here. All surfaces in the oral cavity are coated with a layer of organic material which is known as, and that organic material is known as acquired plaque. Acquired plaque, okay. So first thing is all the surfaces of the oral cavity, both hard and soft tissues, are coated with a layer of organic material known as acquired plaque. Now what is plaque? Pellicle consists of more than 180 peptides, proteins and glycoproteins, including keratin, mucin, prolin, including keratin, mucin, prolin rich protein. So this is protein, okay? This is a short form for protein, okay? Keratin, mucin, prolin rich protein, phosphoproteins, histidine rich protein, and other molecules that can functions as adhesion sites for bacteria. Okay, so what plaque consists of? This acquired plaque consists of more than 180 peptides, proteins, glycoproteins including keratin, mucin, prolin rich protein, phosphoprotein, histidine rich protein and other molecules that can function as adhesion sites for the bacteria. Now the salivary pellicle can be detected on clean enamel surface within 1 minute after their introduction into the mouth of volunteer. See, whenever the plaque is formed inside the mouth of the volunteer, it will detect within 1 minute. Okay, and by 2 hours, the pellicle is essentially in equilibrium between adsorption and detachment. And further, pellicle maturation can be detected in several hours. Okay. Consequently, bacteria does not contact the enamel directly. See, bacteria here does not contact the enamel directly but interacts with acquired enamel pellicle. And many proteins, one more thing you need to know is many proteins retain enzymatic activity when they are incorporated into the pellicle and may affect the physiology and metabolism of the adhering bacterial cells. Okay, so many proteins retain their enzymatic activity when they are incorporated into the pellicle. So many proteins, some proteins I will name, some proteins like peroxidase, lysozyme and alpha amylase. Okay. So many proteins like peroxidase, lysozyme and alpha amylase retains their enzymatic activity when they are incorporated into the pellicle and may affect the physiology and metabolism of the adhering bacterial cells. So these proteins will affect the physiology and metabolism of adhering bacterial cells. So this is all inside phase 1. Phase 2. So in phase 2, initial adhesion or attachment of bacteria occurs. Initial or attachment of bacteria occurs. Okay. In phase 1, what happens is the acquired pellicle formation. Okay. And in phase 2, what occurs is initial adhesion or attachment of bacteria. So, over the first 4 to 8 hours, 60 to 80% of bacteria present are members of genus Streptococcus. Okay. So over the first 4 to 8 hours, 60 to 80% bacteria which is present there are members of genus Streptococcus. Other bacterias that are present at this time are, what all the bacterias are? Obligate aerobes. What are obligate aerobes? These are the organisms that can survive only in presence of O2, oxygen. Okay. Obligate aerobes such as Haemophilus species and Neisseria species. And facultative anaerobes. What are facultative anaerobes? Which are basically anaerobes but can survive in presence of oxygen. So these are the basically anaerobes but they can survive in presence of oxygen. Okay. Such as, example for this is such as Actinomyces species, Veillonella species. So SPP is, see guys, if with any organism SP is written, then it is species, one species. Okay. If SPP is written, then various species, more than one species may be present. So Veillonella, more species, more than one species of Veillonella, more than one species of Actinomyces. Okay. So other bacteria is obligate anaerobes, facultative anaerobes. Now these species are considered as primary colonizers of tooth surface. These are known as primary colonizers of tooth surface. So what are the primary colonizers? The genus Streptococcus bacteria present over the first 4 to 8 hours. Over the first 4 to 8 hours, 60 to 80% bacteria present are 60 to 80% bacteria present are members of genus Streptococcus. Other bacteria obligate anaerobes, facultative, obligated aerobes, facultative anaerobes, and these all are primary colonizers. Okay, or we can say PC. Primary colonizers. They provide, now the primary colonizers provide new binding sites for adhesion of other oral bacteria. Okay, and these primary colonizers, what they do, modifies the local microenvironment of dental plaque biofilm for the survival of other bacteria. Okay, so this we read the primary colonizers modifies the local microenvironment of the dental plaque biofilm for the survival of other bacteria. But how? But the question is how they do this. So by removing oxygen. So for primary colonizers I write PC. Okay, primary colonizers provides conditions that permits the survival of obligate anaerobes. Okay, now phase 3. What happens in phase 3 is secondary colonization and plaque maturation. Okay, so primary colonizers adheres to the tooth surface and provide receptors for adhesion of new bacteria as a part of co-adhesion. Okay, so what primary colonizers do, they adhere to the tooth surface. Okay, first they adhere to the tooth surface and also they provide the receptor for the addition of new bacteria. Okay, they provide the receptor for the addition of new bacteria, and this is what? This is co-adhesion, as a part of co-adhesion, because primary colonizers adheres first to the tooth surface and then they provide a receptor for the addition of new bacteria. So initial colonizers use the available oxygen and causes decrease in the levels of oxygen and favors the growth of gram negative obligate anaerobes. Okay. So what obligate anaerobes are? These are the organisms which can survive only in the absence of oxygen. Okay. These microorganisms which adheres to the cell surface of initial colonizers via a process of co-aggregation are known as secondary colonizers. So these microorganisms which adheres to the cell surface of initial colonizers or primary colonizers via process of co-aggregation, and these are known as secondary colonizers. Now examples of secondary colonizers are such as Fusobacterium nucleatum, Fusobacterium sanguinei, Capnocytophaga species, and P. gingivalis. Okay. So our secondary colonizers does not colonizes to the clean tooth surface or pellicle. So what secondary colonizers does, they does not colonizes to the clean tooth surface or pellicle, but rather they adheres to bacteria that are already in the plaque biomass. Okay. Now secondary colonizers does not colonizes to the clean tooth surface, they or pellicle, they adheres to the bacteria which is already present in the plaque. So what bacteria? The primary colonizers. Okay. Secondary colonizers adheres to the primary colonizers. Now maturation of plaque occurs when gingival margin involves a shift in microbial population from primarily gram positive organisms to high number of gram negative organisms. See how the maturation of plaque occurs. Maturation of plaque occurs when the gingival margins involves a shift in the microbial population from gram positive to the gram negative. Okay, and therefore in later stages of plaque formation, co-aggregation between different gram negative species is likely to predominate. Okay, now one more thing you need to know is corn cob arrangement. Okay, So what is cone-cob arrangement? So long-standing supragingival plaque near the gingival margins demonstrates a cone-cob arrangement. So for example, this is a gram-negative bacteria. Now the outer cocal cells, okay, now the outer cocal cells, which are this is gram-negative bacteria, and these are the outer cocal cells which are firmly attached by interbacterial adherence or co-aggregation. Okay, so the gram-negative bacteria and the outer cocal cells are firmly attached to each other with the help of interbacterial adherence or co-aggregation. Okay. Now we are done with the formation of dental plaque. So if you are interested in more such kind of videos, please do subscribe to my channel and press the bell icon so that whenever I will upload any video related dentistry, you will get notified.